Introduction of beta-catenin. Beta-catenin is a multifunctional protein that plays an important role in the transduction of catenin signals and in the intercellular adhesion by linking the cytoplasmic domain of catenin in the canonical catenin cascade. Beta-catenin is the key factor responsible for transduction of the signal to the nucleus, and it triggers transcription of beta-specific genes responsible for the control of cell fat decision in many cells and tissues. Beta-catenin protein overview. Beta-catenin is an adherence junction protein. Adherence junction, also called the Junula adherens, are critical for the establishment and maintenance of epithelial layer, such as those lining organ surface. Ages mediate adhesion between cells, communicate a signal that neighboring cells are present, and the actin cytoskeleton in ser- serving these roles. Ages regulate normal cell growth and behavior at several stages of embryogenesis, wound healing, and tumor cells metastasis. Cell form and leave epithelia. This process, which involves the disruption and reestablishment of epithelial cell contact, may be regulated by disassembly and assembly of edges, and many also function in transmission of the contact inhibition signal, which instructs cells to stop dividing once an epithelial sheet is complete. Regulating beta-catenin function by phosphorylation. The structural integrity of catenin-based cell junction, and thus the function of beta-catenin, can regulated through phosphorylation by various kinases. Phosphorylating an interaction partner of beta-catenin and thereby introducing a negative charge may increase its binding to the positively charged groove formed by central region of beta-catenin. Three serine residues in the catenin cytoplasmic domain are phosphorylated by the protein kinase. Beta-catenin is an evolutionarily conserved protein. The usually signal beta-catenin gene represents the highly conserved centerpiece within the complex expanded catenin signaling pathway. The catenin pathway features a multitude of paralogous components, especially at the level of catenin and their receptors. The complexity of the level of the catenin in evolution, the catenin genes have been identified which belong to 12 to 30 known different catenin subfamilies, 11 of which are also present in vertebrates, where some additional duplication events within individual subfamilies lead to expression of up to 19 different catenin in mammals. The structure of beta-catenin determines its role and the molecule. The beta-catenin pathway is one of a small set of conserved signaling cascades. Receptor tyrosine kinase mediated pathway that regulate animal development from mammals. Beta-catenins of developmental process is the sum of its dual role signaling and structural, as illustrated by the work of Lanchiko. Mouse embryonic stem cells lacking beta-catenin lost their ability of differentiate into a mesodermal germ layer and do not form any neuroepithelium. Both defects are connected to defective cell junction arising during the differentiation process. Putting back a singling defective beta-catenin restores the integrity of junction. Beta-catenin at the membrane balancing addition and signaling. The majority of beta-catenin is located at the cytoplasmic side of membrane as component of catenin-based cell connection in absence of catenin stimulus. Catenin-based adherence junction contribute of forming polarized epithelial tissue, a characteristic metajon feature necessary for maintaining or organism integrity. Catenins are single-pass transmembrane glycoprotein that engage in Ca2-dependent homotypic interaction with their extracellular region and that also link to beta-catenin through their cytoplasmic tails. Beta-catenin and epithelial transition. An important development process modulated by changes in cell-to-cell adhesion is epithelial transition. During this process, polarized rather immobile epithelial cells change their morphology to highly motile fibroblastoid cells. EMT play an important role during embryonic development and drives cancer invasion and formation of metastasis. One of the hallmarks of EMT is the disappearance of E-catenin from the cellular membrane and its replacement by N-catenin. Canonical signaling in a nutshell. Without a catenin signal, the level of cytoplasmic free beta-catenin are kept low. If not bound to E-catenin, beta-catenin is phosphorylated in the cytoplasm by the activity of a multiprotein destruction complex. Ma- Marking beta-catenin for degradation. This complex consists of the scaffold protein axin and adenoma polyposis coil and of the kinase phosphorylating beta-catenin, glycogen synthase kinase, casein kinase and protein phosphates. The binding of catenin to the fusion transmembrane receptor and coreceptor. It starts a series of molecular events leading to inhibition of the beta-catenin destruction complex. Lysine activated Fraser receptor required. The Nieukoop Center in beta-catenin gene. The Nieukoop Center, named after the development biologist Peter Nieukoop, is a cluster of dorsal vegetal cells in a blastula, which produce both mesoderm-inducing and dorsaling signals. According to the prevalent model of early development, the organizer is induced by the dorsaling Nieukoop signal, which is secreted by Nieukoop center. Formation of the center require the material catenin pathway, which is active on the dorsal side of embryo. Thank you.